Hey, chemists and biochemists. Today, what I'm going to talk about is your positively charged proteinogenic amino acids. Now, I'm going to draw each one of them and introduce you to kind of the main details of these different molecules. All right. So, when it comes to positively charged amino acids, what's important for you to think about in your positively charged proteinogenic amino acids, what's really important for you to think about is I'm specifically talking about by R groups. So I'm talking about my side chains whenever it comes to this classification. Now, when it comes to this definition of positive, well, what we're thinking about is we're thinking about positive at a specific time or something unique. And that unique thing is at a neutral pH. These R groups will have a positive charge. Okay. Now, the molecules, the three proteinogenic amino acids that we're talking about, three AAs, they are lysine, arginine, and finally, histidine. Now, each one of these molecules, lysine, arginine, and histidine, they all have the three-letter codes and one-letter codes. Lysine has a three-letter code of L, Y, S. Arginine has a three-letter code of A, R, G. And histidine has a three-letter code of H, I, S. All three of these molecules also have single-letter codes. They are kind of unique. K for lysine, R for arginine, and H for histidine. Now, of these molecules, the first one that I want to draw is I want to draw lysine. And so the way that I'm going to do that is the way that I draw any and every amino acid. I'm going to start out with my backbone. And I'm going to start out by writing it in the directionality that we read protein sequences. And that is in the N terminus to the C terminus. So the N to C directionality. So what I'm going to do is I'm going to put in the three atoms of my backbone, the three main atoms of my backbone, N, C, C. My N termini, well, that's going to be protonated. Now, I'm going to draw each one of these amino acids at a pH of 1. Then I'm going to draw them at a pH of 7. But first, we're going to go ahead and draw them at a pH of 1. So my N termini is going to be H3 plus. My alpha carbon, my middle carbon here, is going to have a hydrogen coming off of it. And then my C termini is going to also be protonated, and it's going to be a carboxylic acid, COOH. Okay, now my R group for lysine is somewhat simple. It consists of CH2, 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 and then I've got NH3 positive. Okay, now this is my amino acid, lysine, at a pH of 1. I have a total of 3 different ionizable groups, and I'm going to circle all of those. I'm going to circle them in yellow. Well, that's not going to show up well enough, so I'm going to circle them in, let's go with green. My N termini is positively charged and can be deprotonated. My C termini is neutral and can be deprotonated. And my R group is positively charged and can be deprotonated. Okay, that's just to give you a little bit of a consideration of what we're going to be looking at in a little while. Okay, now my next amino acid, I'm again going to draw this in light blue. I've got a backbone. I'm going to draw a little arrow to show where I'm going out here. My backbone is N, C, C. My N termini is NH3 plus. My alpha carbon has a hydrogen on it. And my C termini is COOH. Now, this R group is a little bit complicated. What it consists of is a total of 3 CH2s, making up a chain here. Then what we've got is this kind of complicated functional group that it's important to keep things in mind. We've got a nitrogen going to a carbon, which is double bound to another nitrogen, and that central carbon is also got a single bond to a nitrogen. Each one of these nitrogens off the bottom here have two hydrogens, and this is a resonance stabilized structure. This nitrogen that I've just put a little star by, that I'm going to erase in just a second, let's take stock of what that has with respect to bonds. It has a total of 4 bonds, 2 coming from carbon, and then 1 going to each of these 2 hydrogens. So that nitrogen has a grand total of 4 bonds. And what do we know about nitrogen with 4 bonds? It's going to have a positive charge. Okay, so again, I'd like to highlight, circle my ionizable groups. My N termini is positively charged and is protonated. My C termini is positively charged, it's neutral and it is protonated. My R group, well, I'm going to circle both of these nitrogens because of the fact that it's a resonance stabilized group. We have a positive charge there, and at a pH of 1, it's going to have that positive charge, but that group can be deprotonated. Okay. Now, my last but not least positively charged amino acid is histidine. Now, histidine is a little bit tricky, not so much from the drawing perspective, but instead, well, this kind of classification. Now, histidine, single letter code of H. I'm going to draw this in the same way. NH3 plus C, hydrogen. C double bonded O to an OH. Now, this R group is a little bit tricky because it's going to have some, well, it's a ring structure, and what we've got is a CH2, and then we've got this 5-membered ring. Now, we've got a nitrogen, we've got a double bond going to a carbon, and then we have a H single bond. Okay, we've got a double bond there. We have this nitrogen with a positive charge on it. Okay, now what this ultimately leads to, and what's important to recognize about this, is that this R group is also an ionizable R group. So I'm going to circle my 3 ionizable groups on nitrogen, on this histidine, my N termini, my C termini, and this nitrogen within this ring. That nitrogen can lose a proton, and whenever it loses that proton, it goes from a positively charged R group to an R group with no charge whatsoever. Now, like I said, I've drawn these molecules at a pH of 1. What I want to do now is I want to draw these, not completely redraw them, but make the changes to show these molecules at a pH of 7. Okay, now what I'm going to do is first target my C termini, because my C termini of every single one of these molecules is going to have a pKa of approximately, I'm going to call it 2.1. It's plus or minus a little bit, might range from 1.8 to 2.2, but I'm going to target those groups. So I'm going to erase my green circles. Then what I'm going to do is I'm going to erase my hydrogen on my carboxylic acid, and I'm going to replace that with a negative charge, because that's the first group that's going to be deionized, the first group that's going to be deprotonated. Now I'm also going to change this from at pH 1 to at pH of 7. Okay, now my N termini for every single one of these groups is going to have a pKa of, about 9 or so. So at a pH of 7, they will remain protonated. So I'm going to put a check mark by each one of those. The R group for something like lysine has a pKa of about 10.53. The R group for a molecule like arginine is going to have a pKa of about 12.48. So both of those groups at a pH of 7 will remain protonated. That leaves the R group for arginine. Arginine or I'm sorry, histidine, that leaves the R group for histidine. Histidine is actually going to be deprotonated because histidine has a pKa, or the R group of histidine has a pKa of approximately 6.1. So what that means, let me go ahead and redraw this here. What that means is that histidine, though classified as a positive residue, it's actually only a small fraction of it that is going to be positively charged at a pH of 7. Because if you think about the buffering range of the R group, the buffering range is going to go from 6.1 to 7.1, or actually it's going to range from 5.1 to 7.1. So at a pH of 7, a portion of it is positively charged, a considerably lower portion than something like arginine and lysine's R groups. Okay, so the way that I've drawn all of these molecules as they are displayed now is what they all look like at a pKa of 7. If the at a pH of 7, if the pH of the solution continues to go up, each one of these groups will be deprotonated. But these are our 3 amino acids that are classified as our positively charged amino acids. Now, with them being classified as positive residues, it's also important to think about them as, well, they're polar. They are polar and they are positive. Not all amino acids that are polar are positive, but all amino acids that are positive are polar. At any rate, I hope this was helpful, and I hope this cleared up any sort of confusion that you may have about your positively charged amino acids. All right, have a good one.